moving on. We are doing biomolecules. I'm going through NCERT textbook line by line. Now remember, in my last lecture, please like, subscribe and share my video. I gave you a complete overview of the entire chapter and the concept of biomolecules. One most important thing that I want you all to understand and remember is, we have discussed the concept of acid soluble pool and acid insoluble pool, what is present in the acid soluble and acid insoluble pool. But remember, the NCERT textbook, especially in this chapter, gives you everything in bits and pieces, and I don't want you to go through the entire thing one by one. When you start reading proteins, there is a paragraph here on the second. In my textbook, it's page 105. There is some little detail given about amino acids here. It goes up till here. Zwitterion concept, etc. And then there is a little structure of protein which is given here, which gives you a concept of protein. And the third part is proteins comes here as structure of proteins. I don't want you to start reading everything and get confused. When we deal with proteins, we deal with all these three portions one by one. So I want you to read about amino acids first, structure of proteins second, when it comes to amino acids, and then the secondary, tertiary, quaternary structure of proteins. Similarly, with polysaccharides and carbohydrates, same is the case with lipids. There's a little detail about polysaccharides given here on this page along with nucleic acids. There is a little bit about biomacromolecules given here, and here it gives you about lipids. So please understand, there are three places where proteins are given. There are two places where concepts of lipids are given. So after dealing with this whole chapter, I will run you through the NCERT textbook that gives you where it says protein, you jump on 2 pages and go to the protein. Then you jump another page and go on to the protein. If you don't do that, it will be very confusing for you. So we deal with each of these one by one rather than going through them all together just like your textbook gives. No wonder why the NCERT textbooks get confusing for especially biomolecules for the students. Okay, let's start with the first concept of macromolecules and micromolecules. Just before the structure of protein, they give you a little bit about biomacromolecules and biomicromolecules after the secondary metabolites and primary metabolite portion. This is what I'm dealing with right now. Let's go back to when you treat a living tissue with trichloroacetic acid, CCl3COH. You take a living tissue, whether it is liver, whether it is kidney, whether it is your hand, whether it is your skin, your eye, ear, whether it is a root, whether it is a stem, a bark, a leaf, whatever. Grind it with CCl3COH, which is trichloroacetic acid. And after that, you filter it through a cheesecloth, strainer, whatever. Whatever goes down is called as acid soluble pool, and whatever remains behind is acid insoluble pool. So whatever stays up is acid insoluble pool. Whatever goes down is called as acid soluble pool. Most of the inorganic substances go down in the acid soluble pool, and most of the organic substances stay behind in the acid insoluble pool, which is also called as retentate. What are the organic substances? Well, that are found in a living system because you've taken in a living system. Carbohydrate, fat, protein, nucleic acids, and vitamins, minerals, etc. Now remember one thing. These can also be divided in a second way, in a second method of division. They can also be divided into biomicromolecules and macromolecules. What is the meaning of micro? Small. So molecules that are smaller in size, less than 1000 Daltons, are referred to as biomicromolecules. And those that are less than 1000 Daltons should be this. And those that are greater than 1000 Daltons, this is a measure of weight, are referred to as biomacromolecules. Macro means big. So these are called as biomacromolecules. These are referred to as biomicromolecules. So smaller molecules found in biological systems are called biomicromolecules. Larger molecules found in biological systems are called as biomacromolecules. Simple? Okay. When you talk about carbohydrates, fats, proteins, nucleic acids, etc., three of these, meaning carbohydrates, proteins, and nucleic acids, are polymers. Three of these, listen to this carefully, carbohydrates, proteins, and nucleic acids are called as polymers. Why are they polymers? Because they are built up of several repeating units, and the single unit is called as monomer. Clear? Carbohydrates, Proteins and nucleic acids are polymers, whereas lipids or fats are monomers. They are less than 1000 Daltons. In fact, they are less than 800 Daltons. Then they still are not found in the acid insoluble pool. They are still acid soluble pool. They are found in the retentate. Why is that so? Important 2-marker or 3-marker question. Lipids typically are found aligned with other proteins, etc. They form part of cell membrane where they are present as lipoproteins. Just like go back to my last lecture and refer to this once again. Mostly lipids are found as part of plasma membrane. They are attached to some other component. Lipids are not usually found as single units. They are usually attached in the form of vesicles. And when you with a mortar and pestle, when you break it, it breaks the lipids and they connect themselves with other proteins, etc. around them and form vesicles. Now these vesicles are not easy to pass through a acid insoluble portion. So even though lipids are biomicromolecules, they have a low molecular weight, yet they do not pass into the acid soluble fraction. They stay behind in the retentate or acid insoluble portion. Clear? Is that clear? Lipids, even though they are low molecular weight, they form vesicles when they are crushed. They form vesicles with other molecules, and hence instead of passing on through the acid soluble fraction, they stay behind in the retentate or acid insoluble portion. This can come as a 2 or 3 marker question, and it is also a very important point for you to remember. So now be clear about the difference between biomicromolecules and biomacromolecules. For now, we have covered this portion. Okay, now moving on to the biomacromolecules. The biomacromolecules, the bigger molecules, include carbohydrates, proteins, and nucleic acids. Why did I take these 3 names? Because all these 3 have repeating units of monomers, and they are present as polymers, meaning there are several units of a monosaccharide which combines together to form a large polysaccharide. There are several units of amino acids that combine together to form a large protein. There are several units of nucleotides that combine together to form a nucleic acid. Hence, all these 3 are present as polymers, and they are biomacromolecules, or large or heavy molecular weight compounds. Let's deal with them one by one. What are proteins? Now, in the textbook, it gives us 3 different places. Please go through all the 3 different places, one after the other, rather than leave the pages in between and just go through the protein portion first. What is protein? Protein is the building block of the body. What is the role of carbohydrate in your body? It is energy giving source. What is the role of fat? Again, energy giving forms a part of several organs, organ systems, cell, has an important role to play in enzymes, juices, digestive enzymes, etc. So all these have different roles to play in your body. What is a protein? Protein is a polymer. It is a repeating unit of a single unit which is called as monomer. And the monomer unit is usually an amino acid. Clear? So when a big protein is formed, it is usually repeating units of several single units which are called as amino acids. Is it the same amino acid which repeats itself? No. Different amino acids combine together to form a single protein. There could be 60 amino acids one after the other which are attached together to form a single protein. Several amino acids, remember the word amino acid, which combines together to form a single protein. Now, I will come to what is an amino acid. So, protein is said to be a heteropolymer. Why is it said to be a heteropolymer? That is because the repeating units are not the same amino acid. They may be different amino acids. What are the names of common amino acids? I could tell you a lot of names of amino acids, but what is the meaning of the word amino acid? Amino acid just means something which has amino group on one side, NH2, and a COH group on one side. So what is an amino acid? An amino acid, the name suggests, is a compound which has an amino group at one end, COH group, acid group at the other end. Clear? What is an amino acid? An amino acid is a chemical compound that has NH2 group at one end, COH group at another end. So, for example, if I start describing to you simply a structure of an amino acid, it has H2N. I'm writing it ulta, NH2. Amino group is NH2 group. R is just an alkyl aryl group, whatever. COH is the carboxylic group or acidic group. This is called as an amino acid. This is an amino acid. And how does one amino acid connect to the other amino acid? The other amino acid comes next to it. So supposing COH group comes next to it of the other amino acid, the OH and one H of here hydrolyzes, H2O goes out of the system, HOH, so H2O goes out, and the bond that is left is CONH bond. This CONH link is called as an amide link or a peptide bond. Repeating it again, when two amino acids come close to each other, hydrolysis takes place between the NH2 group of one amino acid, COH group of the other amino acid. H2O goes out of the system. What is left behind is the CONH bond. NH2, one H goes out, so what is remaining is NH here. COH, OH goes, what remains is CO. The CONH bond that is left behind is referred to as the peptide link or amide link. Listen to these words carefully. A protein is formed when several amino acids connect to each other by a peptide bond or an amide linkage. What is a peptide bond or amide linkage? The CONH bond is called as peptide bond or amide linkage. When is it formed? When NH2 group of one amino acid comes close to the COH group of the other amino acid. OH goes out, hydrolysis takes place, the CONH that is left behind is called as amide linkage or peptide linkage. Several amino acids join next to each other like a train ka dabba, one after the other, connected together by the amide bond or the peptide linkage. Are the amino acids in the peptide bond same? No. In the protein? No. They are all different. Which is why a protein is said to be a heteropolymer. It is never the same amino acid which is repeated. How many amino acids are there in nature? Ma'am, there are several amino acids in nature. But the essential ones that are required for all the proteins in our body are 20. So essentially, it is very easy for you to remember the names of all these 20 amino acids. Now you know amino acids, the simplest of amino acid has NH2 group at one end, COH group at the other end. If you replace this R by a simple, say, CH, carbon has valency 4. One is occupied by COH, the other is occupied by NH2. That leaves one carbon and one H. This is the simplest amino acid which is possible. The simple amino acids, like your NCERT textbook gives you structures of these amino acids. Without going through this, understanding of all this would be very, very difficult. Go through these three. So basically, the simplest of amino acids are just amide substituted methanes. So a methane has 4 valency, C, H, 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 H. If you replace one H with NH2, the other H with COH. This is the simplest amino acid which is possible. See, so easy it is to remember the structure of amino acids. Look here, glycine is the simplest amino acid which has C, COOH, NH2, CH, H. If you replace with H with CH3, it becomes alanine. If you replace it with CH2OH, it becomes glycine. These three are the simplest amino acids, and these three are the simplest amino acids that exist in nature. So what are amino acids? They are basically substituted methanes. When you read the structure of amino acids as given in NCERT textbook, they are substituted methanes. They are substituted methanes. So methane, 1 valency, 1, 2, 3, 4. Carbon has 4 valency. There is one H here, one H here. There is a COH here and NH2 here. This is the simplest amino acid, which is glycine. If you replace this H with CH2, it becomes alanine. And if you replace with this with CH2OH, it becomes serine. It is so simple to remember the structures of these 3 amino acids. Yes, glycine, alanine and serine. So what are amino acids? They are basically substituted methanes. Now, when you go to more complex amino acids, the R group here in the amino acid may be an aryl group, so they may be aromatic amino acids. Your NCERT textbook gives you examples of 3 aromatic amino acids, and the 3 aromatic amino acids that is given in NCERT textbook is the 3 are phenylalanine, tryptophan and tyrosine. Phenylalanine, tyrosine and tryptophan are the 3 aromatic amino acids. Based on the number of carboxylic group, if there are 2 COH group, Like in glutamic acid, it becomes an acidic amino acid. Now, I'll introduce to another important concept that NCERT gives. Amino acids also have a characteristic feature, and the characteristic feature of amino acids comes from this. So say NH2 group at one side, which is the amino group, and COH group of the other side, which is carboxyl group. Amino acids also have another characteristic feature. It can either lose a proton here and it can gain a proton here. So an amino acid can exist in two forms. It can exist like this. It can also exist in R NH3 plus, this has gained a proton, and COO minus, which is called as a zwitterine concept of amino acid. It can exist as NH2RCOH, or it can lose a proton. The proton can get transported here. It can become NH3 plus and CO minus. This kind of amino acid can exist in zwitterine form. So they can either gain protons or they can lose protons. And this concept, when an element, when a compound can give proton as well as accept proton, so it can get oxidized as well as reduced, that form is referred to as a zwitterine. And this is a characteristic feature which is found in amino acids. Now keep following, like, subscribe my video.